Welcome to the last part of the respiratory system. Let's take our. We're now going to get into the transport of respiratory gases by blood. Take note, guys, that you have here the red blood cells showing an enlargement of the hemoglobin. That's your hemoglobin. You have the four globin chains, and you have the heme part, those yellow things that look like they have something red in the middle. That's the heme or the red part is the iron, and that's exactly where your oxygen is going to bind. Molecular oxygen is carried in the blood two ways. First, have about 1.5% only is dissolved in the plasma. 98.5% is loosely bound to each of the iron of the hemoglobin, showing right here in the red blood cells. Each hemoglobin molecule is composed of four polypeptide chains, as I mentioned, each with an iron containing heme group. So each hemoglobin can transport four oxygen molecules. Oxyhemoglobin is the result of the hemoglobin oxygen combination. That's the name of the molecule, oxyhemoglobin. Reduced hemoglobin, on the other hand, or deoxyhemoglobin, is this is when the hemoglobin has released the oxygen, and therefore it results in a blood that is not so bright red, still red, but it's dark red. Loading and unloading of oxygen is facilitated by a change in the shape of hemoglobin. As the oxygen binds, the hemoglobin will change its shape, increasing further its affinity for oxygen. The confrontation of the hemoglobin would change. It'll make the globin chains not as tight as before. It will loosen up and it will allow more oxygen to bind to it. But unfortunately, it has only four hemes and only four oxygen can bind to a hemoglobin at the maximum. As oxygen is released, the hemoglobin shape would pretty much cause a decrease in the affinity for oxygen. When the oxygen is going to leave the hemoglobin, the globin chains again becomes tightly bound. When fully saturated, like 100%, all of the four heme groups carry oxygen. Pretty much, the hemoglobin is not anymore going to carry in uh, any more oxygen. A partially saturated hemoglobin is when one or three only, up to maximum of three, would be carried, would uh, would would actually be carrying the oxygen. The association of the oxygen and hemoglobin pretty much is actually the rate of the loading and unloading of oxygen, and is regulated to ensure that adequate oxygen delivery to there's actually adequate oxygen delivery to the cells. There are factors that influence this hemoglobin saturation, such as the pressure of oxygen, and other factors such as temperature, the blood pH, the concentration also, or the pressure of carbon dioxide, and the concentration of a molecule which we call bisphosphoglycerate. And we are going to discuss what bisphosphoglycerate later on. Here's actually the dissociation curve, the oxygen hemoglobin dissociation curve. Now, that's actually a wrong spelling, dissociation curve. This is actually the percent of hemoglobin saturation that has been plotted against uh, the pressure of oxygen concentration. So here's actually the percent of hemoglobin concentration, and this is actually your PO2, or the pressure of available oxygen. The resulting graph is not linear, but is actually like a sigmoid or an S shape. This is referred to as the oxygen hemoglobin dissociation curve. And I mentioned that that's actually a wrong spelling right there. And this is a graphical representation that illustrates the relationship between the partial pressure of oxygen and the percentage of hemoglobin saturated with oxygen. In essence, it shows how readily hemoglobin in the blood cells would bind to and release oxygen depending on the surrounding oxygen's availability. So, let's try to understand the curve, because this curve might be one of the things that you might encounter in your test. For instance, this x-axis is representing the partial pressure of oxygen. That's your x-axis right there. That's your partial pressure. And then the y, the vertical axis, is the percentage of hemoglobin saturated with oxygen. And that's when oxygen and hemoglobin are already like bound together, you call it oxyhemoglobin. So this is actually the oxyhemoglobin percent saturation. And the curve exhibits a characteristic sigmoidal um, sigmoidal shape due to the cooperative binding of oxygen to the hemoglobin. This means that the binding of one oxygen molecule to hemoglobin increases the affinity for subsequent oxygen molecules, which I already mentioned like before. The plateau, okay, the plateau represents the higher peak, and this happens in the lungs. This part here is already the availability of oxygen that is in the lungs. And the curve flattens out would plateau, indicating that even significant increases in the pressure of oxygen is only as will result in only a small increase in the saturation of oxygen in the hemoglobin. That the hemoglobin here at this side here at this point there is already fully saturated in the lungs, allowing for a more efficient oxygen uptake. Here's another way of looking at the association, the oxygen hemoglobin dissociation curve. But it will give you an idea right here of how saturation or how saturated the hemoglobin would be on each part of the curve. Now, this will also tell you that, like up there at that particular peak where you have the plateau, your arterial blood at that point here is already pretty much like the pressure of oxygen is about 100 uh, millimeters of mercury, and it contains 20 ml of oxygen per 100 ml of blood. That's exactly what it means. But oxygen at this point in time is by volume is 20 volume percent, and the hemoglobin is 98% saturated here, which means 98 or 100%. You could see one hemoglobin there, and it will have 4 oxygen that has already filled it. Now, uh, if you will be doing like heavy maximal breathing at this point in time, it will not cause any increase in oxygen binding because it has already reached its maximum point. One hemoglobin molecule can only carry 4 oxygen. And it will show you that for that right here in what do you call this, in the lower portion and in the venous blood right there, let's just say 40 right there, where you have 40 millimeters of mercury pressure, it is actually containing only 15% volume of oxygen. This is actually where your venous blood is, right around this area here. And your, well, actually, No, not here, because this is already like 50%. And when you have 50% of oxygen in your hemoglobin, uh, you are not anymore going to be able to breathe. You're you're pretty much like going to um, lose consciousness already at this point in time. This point here this point here is actually the point where you have arterial blood. Let me see this because it's really tiny. Um, in the tissues and in the organs where PO2 is, there you go, where you have 40 millimeters. Okay, all right. This is actually the spot. There you go. This is the spot where 40 millimeters of mercury pressure. I'm looking at the wrong axis right there. So this is actually a 40 millimeters of mercury pressure where you have 75% of hemoglobin is still available, and that is already in the venous blood. That means you have already exhaled air, and yet still, take a look at how versatile your hemoglobin is. It has only lost, for every hemoglobin that's around, it has only lost one oxygen. So each hemoglobin molecule is still 75% saturated in the venous blood. This would allow for whenever there's a possible need for more oxygen, whenever there's actually low oxygen in the atmosphere, or probably you are in an area where oxygen is very, very low, then you know that you will be gasping for air, but you know that your venous blood still has 75% oxygen right there. Okay. And over here in this particular sigmoidal dissociation curve, it shows us that um, at high pressure of oxygen, there are large changes in the, um, the pressure that is caused by small changes in hemoglobin saturation. Notice that the curve is relatively flat right on this part here. The curve is relatively flat. There is pretty much let me see, the hemoglobin properties produce a safety margin pretty much that ensures that the hemoglobin is almost fully saturated even with a substantial change in the pressure of oxygen decrease. As a result, the hemoglobin remains saturated even in high altitude or with lung disease. And just like what I mentioned to you, whenever you are in need of oxygen, whenever you are in need of oxygen, such as when you are in high altitude or when you are swimming, that particular 75% saturation of hemoglobin will be able to compensate for your tissue need for just in case there's actually a big drop in the oxygen. So there are other factors that influences this particular curve. Um, the amount of oxygen that's carried by hemoglobin depends on the pressure of oxygen or the amount of oxygen available locally. And this relationship ensures optimal oxygen pickup and delivery. And let's take a look at the effect of exercise. Um, okay, before we get into the exercise, I mentioned to you that I'm going to explain to you or mention to you what BPG is because this pretty much I saw this particular thing that says 2,3-BPG. It is actually referring to the biphosphoglycerate, which our red blood cells produce. That is actually one of the byproducts of our glycolysis whenever we're actually undergoing metabolic um, aerobic reaction. So BPG levels rise when oxygen levels are low. And as cells metabolize glucose, they use the oxygen, causing increase in the pressure of carbon dioxide and hydrogen ion in capillary blood. The declining pH, which results in acidosis, and increasing pressure of carbon dioxide would cause the hemoglobin and hemoglobin oxygen, that's actually oxyhemoglobin bond, to weaken. Now, this weakening effect is what you call the Bohr effect. Any factor that will cause to the weakening of the hemoglobin bond is actually exhibiting Bohr's effect. Hence, when the bond is weakened, the oxygen unloading would occur where it's actually needed the most. Heat production in active tissues directly and indirectly decreases the hemoglobin affinity for oxygen. It allows increased oxygen unloading only to the active tissues. There are other factors also that affect hemoglobin saturation. Example of this, that's like recorded here. You have an increase in temperature, hydrogen ion or the presence of the hydrogen ion, the pressure of carbon dioxide or the presence of carbon dioxide, BPG, and these can modify the structure of hemoglobin. All of them result in the decrease for hemoglobin's affinity for oxygen. They occur in systemic capillaries. They enhance oxygen unloading. They cause a shift in the oxygen. Take a look at this dissociation curve to the right. All of this is causing the dissociation curve to the right. Now, I have here a huge red blood cell. This huge red blood cell is within a capillary, and these are actually your tissue cells. This is your capillary cell, and this is the interstitial fluid, where pretty much there is actually an exchange of gases, substances, and gases, depending on the availability. Remember that the movement here is not anywhere close to being like active transport. No, this is passive transport. The transport of gases is dependent on concentration gradient. If you have too much of this gas on one side, it moves to the opposite side. If you have too much of this other gas on this side, it moves to the opposite side. That's the only drama here. There is no other reason why these substances will actually exchange places. Both oxygen and carbon dioxide, or the pretty much the major gas players in the body, are pretty much like doesn't need any carrier to pass through our membranes. They are pretty much able to pass through with ease. Carbon dioxide is transported in the blood in three forms. They could be dissolved in the plasma, right there. Carbon dioxide from the tissues. You have too much carbon dioxide. You produce carbon dioxide in glycol in what do you call this in what do you call in your production of ATP. One of the end products is carbon dioxide. That's actually in the Krebs cycle, and carbon dioxide will pass through passively. Okay, it's only by passive transport wherever there's more of them into right into the blood, and they are dissolved in the plasma. About 7 to 10% of the carbon dioxide is dissolved in the plasma, and then they could also be chemically bound to hemoglobin, and just over 20% of the carbon dioxide could be bound to hemoglobin. Carbon dioxide is bound to the globin part of the hemoglobin and is referred to as the carbamino hemoglobin. Is the binding of carbon dioxide to the hemoglobin does not have a tight affinity. They're really very loose. You could easily lose the carbon dioxide. So only 20% of that is bound to hemoglobin, the globin part, not in the heme part. As your um, as bicarbonate ions, These are your bicarbonate ions right there, bicarbonate ions there. About 70% of carbon dioxide is converted into carbon dioxide is converted to slowly converted into bicarbonate ions. It ionifies itself into hydrogen ion and the bicarbonate ions. Pretty much this is um, the formation of bicarbonate involves carbon dioxide combining with water. There you go. There you go. To form carbonic acid, which is this one, which pretty much quickly dissociates into bicarbonate and hydrogen ion. This occurs primarily in the red blood cell, where the enzymes carbonic anhydrase is reversibly and rapidly catalyzing this reaction. The reaction is there, it's right here also. This is actually the carbonic anhydrase. And that's the reason why this is a very fast, a very quick reaction, because it has an enzyme that catalyzes it. This is actually inside the red blood cell. In the systemic capillaries where the bicarbonate ion is created, this is actually in the capillaries where the systemic ion is created, it quickly diffuses from red blood cells into the plasma, outside into the plasma. The outrush, the outrush of the bicarbonate. There you go. You have up there. That's the outrush. The outrush of the bicarbonate ion from the red blood cell is balanced off as chloride moves into the red blood cell from the plasma, and it moves right here. Okay, this is referred to as the chloride shift. The movement of your chloride from the plasma into the red blood cell is your chloride shift. In pulmonary arteries, now this is now the case where you have the alveoli on the side. This is not any of the tissues. And this is now your blood. You're now carrying the waste gas from the blood into the alveolar sac. The percentage or the presence of carbon dioxide here is much lower than the blood. So the movement of carbon dioxide, because you're carrying a lot of carbon dioxide from the tissues, when you reach the lungs, all of them will be driven out by passive transport into the alveolar sac. The bicarbonate, this is actually through the movement of your bicarbonate. The bicarbonate moves into the red blood cell while the chloride moves out of the red blood cell back into the plasma. And the bicarbonate ion binds with the hydrogen ion to form again the carbonic acid. This is H2CO3. The carbonic acid is split by the carbonic anhydrase there, right there down here, by the carbonic anhydrase into carbon dioxide and water. Carbon dioxide readily will be diffusing out from this side of the wall of the alveolar sac to the inside of the alveolar sac, and then because that eventually will increase the concentration of carbon dioxide here, it will have to go and move out. Concentration of carbon dioxide inside your lungs from this side here to 40, it will move here, and this becomes 40 milliliters of mercury pressure. It will readily move out because the concentration of carbon dioxide in the air is less than 40. So let's take a look at the Haldane effect. What's the Haldane effect? The Haldane effect describes how the affinity of hemoglobin for carbon dioxide is influenced by the oxygenation state of the blood. More specifically, when hemoglobin is deoxygenated, as in deoxygenated in the tissues, its affinity for carbon dioxide increases, allowing it to pick up more carbon dioxide from the surrounding cells. So the hemoglobin as the hemoglobin releases the oxygen, its affinity for carbon dioxide now is going to be increased. And therefore, that's the time when the carbon dioxide from the tissues will now be readily bonding to the hemoglobin. When hemoglobin is oxygenated in the lungs, its affinity for carbon dioxide, this is actually the reverse, is now going to decrease, facilitating the release of carbon dioxide into the alveoli in order that it will be released outside, or it will as you breathe it out. This process would enhance the transport of carbon dioxide in the blood and works in conjunction with the Bohr effect, and which describes how CO2 and hydrogen ion affects hemoglobin's oxygen affinity. That's actually to ensure efficient gas exchange in the lungs and the tissues. So the affinity of the oxygen for both the carbon dioxide and the oxygen changes as it goes to the lungs and in the tissues. In the lungs, it will need to release the carbon dioxide and would increase its affinity for oxygen because that's when it really receives also the oxygen. In the tissues, it will increase its affinity for the carbon dioxide. In the lungs, it will lose its affinity for carbon dioxide because it is not much needed. It will have to lose the carbon dioxide and take in the oxygen. It will increase its affinity for oxygen. In the tissues, it will decrease its affinity for oxygen so it can easily unload the oxygen to the tissues. It increases its affinity for carbon dioxide so it can pick up the carbon dioxide in the tissues. So let's take a look at the influence of carbon dioxide on blood pH. Um, Pretty much by understanding the carbonic acid bicarbonate buffering system. The the carbonic acid buffer system is the primary buffer system in extracellular fluid, especially in the blood plasma. It's essential for maintaining blood pH when a narrow range, within a narrow range of the blood pH is only 7.35 to 7.45. So it's crucial for the proper functioning of enzymes and metabolic processes. This reaction is catalyzed by the enzyme carbonic anhydrase, making it one of the fastest enzymes known in the human body. It's actually called carbonic anhydrase. The equilibrium shifts to the right towards the high, to, the what towards the right towards the bicarbonate and hydrogen ions, towards the hydrogen ions and bicarbonate. Okay, when there is an increase in carbon dioxide. So whenever you have carbon dioxide, this would be produced, carbonic acid, and then it's going to go immediately towards the right. That is during metabolism. Conversely, if the hydrogen ion concentration increases, if this hydrogen ion concentration increases and lowering the pH, the bicarbonate reacts to form carbonic acid, shifting the equilibrium to the left. Okay, this picture shows us changes in respiratory rate and depth as it affects blood pH. Slow, shallow breathing causes an increase in carbon dioxide in the blood, resulting in a drop in the pH. And this is actually where you're not breathing too much, you're hypoventilating, okay? You are increasing your carbon dioxide level, and therefore you are increasing the acid, that is actually hydrogen ion that is being produced by the carbonic acid, causing a drop in the pH or resulting in a drop in the pH. Now, on the other hand, hyperventilation, deep breathing, causes a decrease in the carbon dioxide in the blood. Here you're actually releasing a lot of the carbon dioxide, resulting in a rise in the pH. Changes in ventilation can help adjust pH when disturbances are caused by metabolic factors. Hypoxia is a condition when there is a deficiency of oxygen in the body's tissues, potentially affecting the entire body or just specific regions. It can arise from various causes, including breathing problems, environmental factors, toxicological issues, and even neurological conditions. Hypoxia can be a serious medical concern, leading to symptoms like confusion, difficulty in breathing, and rapid heart rate, and can be even life threatening. Anemic hypoxia. Anemic hypoxia is when there's too few red blood cells or the red blood cells are abnormal and so there's actually maybe too little hemoglobin, and you are always like gasping for going to be gasping for breath or in need for breath. You are going to be hypoxic because you are anemic. Ischemic hypoxia, number 2, is the impaired or blocked blood circulation, resulting in poor circulation of gas where it's actually needed. Histologic, histologic hypoxia. The cells here are unable to use oxygen, as in the presence of metabolic poison like cyanide. This will result in inability of the cells to use up oxygen. Then you have also your hypoxemic hypoxia. This is the second to the last right there. This is because of abnormal ventilation or pulmonary disease or low levels of oxygen in the air. And finally, the last one is your carbon monoxide poisoning, especially from fire. The hemoglobin pretty much has an affinity. Hemoglobin has 200 times greater affinity for carbon monoxide than oxygen. Sadly, that's actually how carbon monoxide would kill us. It will, because it has more affinity in hemoglobin, it easily could get into the hemoglobin, and pretty much your oxygen will not have any place to bind. Carbon monoxide is odorless and very hard to trace. Victims will have headaches and can become flush and requires several days to recuperate, if at all they will not get killed because of the poisoning. Now let's take a look at respiratory rhythms. Respiratory rhythms are regulated by higher brain centers, chemoreceptors, and pretty much other reflexes. You can see it here. Um, the breathing is actually regulated here. Right there, you have your respiratory centers right there. And you have also the chemoreceptors that is found in the aortic sinuses. The ventral respiratory group. Where is your ventral? There we go. Ventral respiratory group and dorsal respiratory group. Where is it? Right here in the picture. Ventral respiratory group, right there. And then the dorsal respiratory group will be right there. So what are these? Ventral respiratory group, or VRG, is the rhythm generating and integrative center. Recent evidence actually suggests that forced expiration may involve a separate group of neurons than passive respiration. These VRG consist of neurons or network of neurons in the brain stem that extends from the spinal cord to the pons and medulla junction. It sets eupnea or normal respiratory rhythm to about 12 to 15 breaths per minute. Its inspiratory neuron will excite the inspiratory muscle. The inspiratory neuron will excite the inspiratory muscle via the phrenic nerve in the that goes in the diagram, from the diagram, and the intercostal nerves. The expiratory neurons inhibit the inspiratory neurons. On the other hand, the dorsal respiratory group here, DRG, this is a network of neurons that's located near the roof of the cranial nerve number 9, and the group integrates input from the peripheral stretch and the chemoreceptors that sends information to the ventral respiratory group neurons. And pretty much up to now, there's not really much known fact, or not too much information is readily available about this group of neurons and its effect on respiratory rhythm. So let's take a look at the factors that affect our breathing. First of all, let's take a look at chemical factors, which is actually the most important of all factors affecting the depth and the rate of inspiration. The changing levels of carbon dioxide, oxygen, and the pH going up and going down are the most important. Levels of these chemicals can be sensed by the central chemoreceptors, and the central chemoreceptors that is located in the brainstem. So this is actually your brainstem. You have chemoreceptors. You got actually stress receptors here, and you have chemoreceptors, and there are also peripheral chemoreceptors right there that is located in the aortic and carotid arteries, and this would affect our breathing. The pressure of carbon dioxide, pretty much most potent and most closely controlled. If the blood carbon dioxide levels would pretty much like rise. Carbon dioxide accumulates in the brains and pretty much joins with water to become carbonic acid. And this carbonic acid dissociates and releasing hydrogen ion, causing a drop in the pH, just like what I mentioned already. An increase in the hydrogen ion stimulates the central chemoreceptors of the brainstem, which synapses with the respiratory regulatory centers. The respiratory centers would increase then the depth and rate of breathing, which acts to lower the blood carbon dioxide level, and pH rises to normal levels. If the blood carbon dioxide level would decrease, on the other hand, inspiration becomes slow and shallow, and apnea may result, meaning breathing may stop because the carbon dioxide level drop abnormally low. Swimmers usually sometimes voluntarily hyperventilate to enable them to hold their breath longer, causing a drop in the levels of carbon dioxide in their blood, which causes a delay in respiration. As the presence of carbon dioxide, or the levels of carbon dioxide need to build up back, and that's actually a technique that's actually being done by swimmers. It can cause a sudden drop, can cause dangerously drops also in the oxygen level. What about oxygen? What's the influence of the presence of oxygen? You have peripheral chemoreceptors right there in the aortic and carotid sinuses. They sense arterial oxygen levels, and declining oxygen levels normally has only slight effect on the ventilation because of the huge oxygen reservoir that is bound to hemoglobin. Even if you get rid of the oxygen, you exhale the oxygen out, that you're only exhaling 25% or one of the oxygen out of the four oxygen that is already in the hemoglobin. So your hemoglobin pretty much has 75% oxygenation still. There should be a substantial drop in the arterial oxygen, pretty much below 60 millimeters, instead of just 75, below 60 millimeters, to stimulate an increase in ventilation. When excited, the chemoreceptors will cause the respiratory centers to increase ventilation. Now, arterial pH. Let's take a look at the influence of arterial pH. pH can modify respiratory rate and rhythm as well, even if carbon dioxide and oxygen levels are normal. They are mediated by peripheral chemoreceptors. And a decrease in pH may reflect carbon dioxide retention or accumulation of lactic acid or excess ketone bodies. Too much exercise can actually do that. Respiratory system controls attempt to raise the pH by increasing respiratory rate and depth. And therefore, if there's actually like very low pH because of what's going on metabolically in your body, you will feel the need to uh, hyperventilate. So in summary, let's take a look. In summary, let me just say something in summary. Rising carbon dioxide levels are the most powerful respiratory stimulant. Normally, your oxygen availability affects breathing only indirectly by influencing peripheral chemoreceptors' sensitivity to changes in carbon dioxide. When arterial oxygen falls below 60 millimeter, it becomes major stimulus for respiration via the chemoreceptors. So changes in the arterial pH resulting from carbon dioxide retention or metabolic factors act indirectly through these peripheral chemoreceptors. So the chemoreceptors are really very vital in the way we're actually responding to changes in these chemical chemical things brought about by the presence of the gases in our body. These chemoreceptors can help detect it and can increase or lower down the depth of our breathing or the uh, um, the availability because of the availability of gases. Now let's take a look at besides oxygen and carbon dioxide and the acidity of the blood or the pH of the blood. There's also other uh, things that can influence our breathing, and that's the influence of the higher brain centers. The hypothalamus, somewhere in our hypothalamus, um, acting through the limbic system, the hypothalamus modifies also the rate and depth of respiration. For example, breath holding that occurs because of anger or gasping with pain would change, and also change in body temperature changes the rate of respiration. Cortical controls from your cerebral cortex. Cortical controls direct signals from the motor cortex that bypasses medullary control. For instance, voluntarily holding your breath until at least the brainstem will reinstate breathing when blood carbon dioxide becomes critically high. Okay, when you are actually holding your breath, as in when you're swimming or playing a game of holding your breath, you cannot hold it for so long. You can only hold it for so much because when carbon dioxide level becomes critical, you will have to exhale it. There's nothing you can do. Or there are some other pulmonary irritant reflexes. Receptors in the bronchioles respond to irritants such as dust, accumulated mucus, or nauseous fumes. These receptors communicate with respiratory centers via the vagus nerve and they promote reflexive constriction of air passages. Some irritants will trigger a cough reflex in the trachea or bronchi or sneeze in the nasal cavity. These are non breathing uh, movements, but they are part of our respiratory interactions. There's another reflex here. As a matter of fact, when I was studying anatomy back in the old days, um, we started, I at least, we um, we started our respiratory system when our professor asked us, "Do you know how to breathe? Why you breathing in and why you breathe out? Why is it that you can only hold your breath so much? You can only take in. How is it that you are able to take in air, and after a while you need to take out air?" Well, the answer to that, one of the answers to that, of course, we know already of the interactions between the gases and acidity, and those all of these reflexes is also what you call. There's also what you call the inflation reflex. When you inhale and increase inflation, there's going to be certain point that you're going to stretch out too much your lungs, and that's going to stimulate your stretch receptors in the lungs. And when your stretch receptors are already stimulated, they will say, "Oh, you are already overstretching the lungs. Let me deflate it." That's actually what you call the Herring Brewer reflex. And is a crucial reflexive mechanism that helps regulate the depth and the rate of breathing, protecting the lungs from overinflation. It is triggered by the activation of pulmonary stretch receptors located in the walls of the bronchi and the bronchioles within the lungs. And these receptors are sensitive to mechanical stretching of the airways during inspiration, particularly during deep breaths or prolonged inspiration that exceeds your normal tidal volume. Stretch receptors in the pleura and airways are stimulated lung by lung inflation. They send inhibitory signals to the medullary respiratory centers to end inhalation and allow inspiration, and may act as protective response more than as a normal regulatory mechanism. Let's take a look at our respiratory adjustments. What our body is actually doing to, to adjust for some crucial changes outside or inside, like sort of like a homeostatic response, something, in order to maintain balance. Now, you have here three things: passive exposure to ambient hypoxia, and that's actually because of altitude, because you have low oxygen levels in high altitude. Then you have physical exercise. You are actually consuming your oxygen faster than usual because you need it in the production of ATP. Then you have your combined exercise as well as ambient hypoxia. Let's take a look at this. Exercise first. During exercise, adjustments geared to both the intensity and duration must be made by the body. In the hyperpnea, which means there's going to be hyper or increased ventilation in response to metabolic needs. That's why when we exercise, we pretty much like will be breathing faster than usual. The ventilation here can increase 10 to 20 fold depending on your oxygen need. Ventilation pretty much would increase abruptly. It only increases gradually first initially, then increases at a steady state. But when exercise stops, there is a small abrupt decline in ventilation, followed by a gradual decrease. Uh, Pretty much the pressure between of carbon dioxide and oxygen and pH remains surprisingly constant during exercise. An abrupt increase in ventilation that occurs as exercise begins involves three neural factors. First, you have a psychological stimulus, and which is the anticipation of the exercise, which is simultaneous cortical motor activation of skeletal muscles and respiratory centers. The excitatory impulses to the respiratory centers from the proprioceptors in the moving muscles, the tendons, the joints. There's actually gradual increase and plateau of respiration reflex and is caused by increased rate of carbon dioxide delivery to the lungs, and hence ventilation declines suddenly as exercise ends because the three neural factors are shut off. The gradual decline to the baseline is due to decline in carbon dioxide flow after exercise ends. Exercise leads to anaerobic respiration and the formation of lactic acid. The lack of oxygen in the muscle is not from poor respiratory function, but rather from insufficient cardiac output or skeletal muscles' inability to increase oxygen uptake. Now, let's go to a quick travel to altitude about 2,400 meters, about 8,000 feet. This may trigger symptoms of acute mountain sickness. Oh, I know how this is. I went to the Alps about a few months ago. In March, I went to the Alps, and pretty much we went up to an elevation of 3,000, more or less, and I had mountain sickness. Boy, that is not really pleasant. The atmospheric pressure or the presence of carbon dioxide levels are lower at higher elevation. And as you go higher, you can feel it. And no matter what you do, every single step, every single movement, every single thing, even if it's only like a step that you're actually doing, you are actually going to breathe three times. And yeah, true. Symptoms will be headache, shortness of breath. You're going to be nauseous and dizzy, and in several cases, lethal cerebral as well as pulmonary edema. I did not last long in that. I had to go down to the level where I'm more comfortable because I know what's going to happen next. I am too old for that, but it was really fun. I had fun in the Alps. So our body actually um, can acclimatize. The respiratory and hemopoietic adjustments are made with long-term moves to high altitude. If you're going to live right up there in the high altitude or in the Alps, uh, you can. I can see a lot of people really like doing their regular job there, and they're really very active. Of course, they're younger than me, being like 74 years old and going up there pretty much is really it's very tedious to my breathing. Chemoreceptors, though, become more responsive to the level of carbon dioxide and oxygen as they decline. Substantial decline would directly stimulate the peripheral chemoreceptors, resulting in in minute ventilation that stabilize within a few days, about 2 to 3 liters per minute, higher than at sea level. A high altitude conditions always result in lower than normal hemoglobin levels saturation due to the availability of less oxygen. And oxygen stimulates the kidney at this point in time to accelerate, and this is the better part, to accelerate the production of EPO. And you know what EPO is, right? Erythropoietin. This is now going to get your bone marrow to produce more red blood cells. Red blood cell numbers will slowly increase to provide long-term compensation for the inability of the hemoglobin to take in the oxygen. Then more red blood cell will have to be around to take in the right amount of oxygen level that is needed by the body to survive. Okay, that's pretty much like the end of this particular chapter. your assignment will be to read on lung diseases in your textbook. We have also a video of lung diseases that's available to you in the same lecture series that you saw this video on. And it's right there. You just have to click on it. It's not mine. It's actually something that is available to us. Click on that and um, listen to it. It's a video, or you can read. You can read this in your textbook. It's available in your textbook. I'm going to see you guys in the next chapter.